Hello, ladies and gentlemen. I welcome you to this year's summary of the European School of Hematology's meeting on chronic myeloid leukemia, which took place in 2025 in beautiful Estoril in Portugal. I would like to present to you the biological highlights of this meeting. Once again, it was a terrific meeting full of very insightful information. First of all, there were some lectures on the leukemic stem cells. As you know, these are the cells that drive relapse of chronic myeloid leukemia. Investigators found that there's a skewing of leukemic stem cells towards the erythroid and the megakaryocytic lineages, and it seems as if CD90 is a marker that characterizes leukemic stem cells better than any of the previous ones. In fact, high expression of CD90 seems to be associated with better outcome. And it seems as if there are certain inflammatory signatures in these leukemic stem cells, for instance, with regards to cytokines, megakaryocytes, erythrocytes, as just mentioned, that seem to be playing a role in the response of patients to therapy. In fact, in patients with blast crisis, it seems as if there are very high levels of inflammation within these within these leukemic stem cells. Inflammatory leukemic stem cells and megakaryocytes, in fact, may be the drivers of CML recurrence. There was another talk about Interferon-α. As you know, Interferon-α was previously used to treat CML many years ago. There seems to be a very strong effect also on mesenchymal stromal cells, which are cells within the bone marrow microenvironment, and on the secreted factors by these mesenchymal stromal cells, on metabolic factors within these cells, and on components of the extracellular matrix. Interferon acts via interferon-stimulated genes, or ISGs, and these are actually highest in normal hematopoietic stem cells, less present in more mature cells. It seems as if these high and low interferon-stimulated genes, high and low, are functionally distinct. And it seems as if one is looking for interferon-responsive cells in CML, it seems as if these low ISG cells are actually the driver of CML expansions. And nilotinib actually decreases the expansion of these interferon-stimulated genes low cells. ISG low cells also have a more aggressive outgrowth of the CML. There was a talk, or two talks actually, on new models of CML. There's a new mouse model of CML using an inducible, a different promoter than in the past. These mice, when they have the disease, actually have a 300-day survival with CML, which seems to resemble the human situation and the more stepwise progression of the disease much more closely. There was a new model presented on induced pluripotent stem cells from patients with CML, and with these cells it is possible to study now the good versus the poor responders to tyrosine kinase inhibitor treatment. There were more talks on the microenvironment, as you know, which is the environment that surrounds the leukemic cells in the bone marrow, and it was found that there are specialized macrophages in the CML bone marrow microenvironment, which are positive for certain markers, which decrease with tyrosine kinase inhibitors, and they change also in their phenotype in response to tyrosine kinase inhibitors. It seems as if these CML macrophages polarize differently and interact quite differently with the normal hematopoietic stem cells. And we know that these CML macrophages support the leukemic stem cells in CML, but if you have TKI treatment on board, these macrophages actually support the normal hematopoietic stem cells. It was also shown that depletion of these macrophages reduces leukemic stem cells in CML. Another talk actually presented that megakaryocytes are the cells that drive myelofibrosis in CML. More talks were given on metabolism, really presenting that there are distinct metabolic profiles of tyrosine kinase inhibitor sensitive versus resistant cells. We know that TKI sensitive cells have reduced oxidative phosphorylation, increased mitochondrial DNA mutations, whereas TKI-resistant cells have a remodeling of the mitochondria. These are these little organelles within the cell that drive the metabolism. There was a fascinating talk on the molecular pathology of chronic myeloid leukemia, suggesting that there is not just one translocation, namely BCR-ABL, in chronic myeloid leukemia, but it seems as if there are several. If you have a breakage of a chromosome that leads to the development of the Philadelphia chromosome, characterizing CML, the chromosome actually doesn't just cleanly break, it actually shatters, leading to the formation of many, many complex, small little translocations, all of which seem to contribute to disease development and specifically also the response to treatment. We heard some talks about blast crisis and specifically the epigenetic features of blast crisis, and we were told that certain transcription factors are responsible for leukemic stem cell features such as AP1, NF-kappa B, and STAT5 also play a very important role in blast crisis. We heard an excellent talk about cell therapy options, not just in CML or specifically in other hematological diseases, and trials on natural killer cells were presented, or for instance, new chimeric antigen receptor T cells or NK cells specific for these diseases that are positive for the marker CD19, such as acute lymphoblastic leukemia. We heard about forms of resistance to tyrosine kinase inhibitors, specifically about certain isoforms of the BCR-ABL fusion protein, and we know that certain isoforms that miss the A2 domain within BCR-ABL, which harbors the SH2 domain, are actually resistant to asciminib. And of course, there are other mutations also leading to resistance in treatment with asciminib. We know that resistance often arises in patients with certain mutations, such as ASXL1, which is a mutation that is associated with aging, and we were told that some of these ASXL1 mutations may have something to do with another metabolic pathway involving ALOX5. There were talks on new drugs, which were very exciting. One of the new drugs is ELVN-001, which is a highly selective ATP competitive inhibitor of BCR-ABL, which had a favorable safety and tolerability profile. We heard about STAT5 nanobodies, which are much, much smaller than antibodies and lack some of the essential domains of antibodies, but these seem to be able to decrease the viability of BCR-ABL positive cells. And very excitingly, we heard about BCR-ABL degraders. These are drugs that can bind to BCR-ABL, and instead of inhibiting the kinase domain, such as in tyrosine kinase inhibitor, these degraders can actually degrade the target protein and thereby reduce the very early leukemic stem cells, at least in vitro. We had a very exciting symposium on myeloproliferative neoplasia, studying fibrosis in mice that is driven by certain proteins such as JUN, then as well as by cytokines, for instance, interleukin-6. We heard about the biology of mutations, such as mutations in calreticulin, that lead to the development of myeloproliferative neoplasia, and we heard about new treatments for myeloproliferative neoplasia, to which chronic myeloid leukemia belongs, such as antibodies to the mutant calreticulin or BET inhibitors. We also heard about new biomarkers in MPN. Once again, we had two terrific workshops on the biology or new methodologies that can be used to study chronic myeloid leukemia and also many other hematological cancers and solid tumors. One of the workshops was on mitochondria and metabolism. We heard about the role of mitochondria in neurodegeneration, cancer, autoinflammation, and we also learned that mitophagy, actually the process of the cell eating certain organelles, in this case the mitochondria, might explain diseases such as Parkinson's disease. In fact, it was also shown that there are mutations that can also occur not just in the whole genome of the cell, but also in the mitochondria, which also have some genes. And these are usually genes in the respiratory chain, so basically in those compartments that drive the metabolism of a cell. We also know that mitochondrial metabolism plays a role in aging of hematopoietic stem cells, and that the membrane potential of these mitochondria drives DNA repair, etc. We also had a very exciting workshop on clonal tracking and lineage tracing. This really allows tracking which malignant clones are present at an early age and then further develop and become sort of monoclonal, the only clone that can give rise to a disease, and in particular a hematological cancer. You can track the acquisition of certain mutations, such as mutations in p53, a tumor suppressor gene, and you can do this in various diseases and clinical settings, for instance, after hematopoietic stem cell transplant. It was shown that TET2, a gene which is frequently mutated as we all age, actually is a super competitor. So if you have a mutation in TET2, it seems to outcompete many other clones and can drive the development of a hematological malignancy. And in another talk, it was shown that you can also do clonal tracking by epigenetic changes. So these are changes that occur as life goes on in every one of us. We heard about the John Goldman Research Award, in which our colleague received an award to study the observation of and the detection of tyrosine phosphorylation after just one tablet of a tyrosine kinase inhibitor. And this technique that is used for this is mass cytometry, and we learned that this is actually predictive for response. We heard about the Janet Rowley Prize, which this year was given for the study of the immune landscape in CML and the response to treatment, and how this was used to predict treatment-free remission and understand the involvement of natural killer cells. It was shown how you can manipulate these natural killer cell receptors and how natural killer cell can lead to resistance or sensitivity to tyrosine kinase inhibitors. And as to miscellaneous talks, we heard about mathematical modeling and in metastatic cancer. We heard about different proteins responsible for the unfolded protein response. We heard about in silico modeling of the kinase domains and different ways to detect biomarkers in CML. Finally, the last talk was by the Max Foundation program, which manages to detect BCR-ABL in dried blood spots, for instance, in low-income countries. And then these cards containing the dried blood spots can be sent across to North America or Europe, where then sophisticated technologies such as next-generation sequencing can be performed. That's it from me, and I think we can thoroughly look forward to the John Goldman Conference next year, 2026, where we will be telling you about more that's been happening in CML. Thank you very much for your attention. Bye-bye.